Well, good afternoon, everybody. My name is Jesse, and I am with Exploring by the Seat of Your Pants. If you're joining us for the first time, we're all about bringing conservation, adventure, and science into classrooms around the world. And a big welcome to our new audience today. I know we've got some live folks in West Kelowna and people all over British Columbia. And so welcome to the broadcast. We do about 40 of these every single month. And this one, like our entire Gardner series, which I'll highlight in a minute, are all on our YouTube channel. Over 2,500 past broadcasts over the last 7 years, if you're interested. Now, today marks day 3. Yesterday, we had to postpone it, actually. So day 2, officially, of our Gardner Foundation celebration of some really, really amazing biomedical and health researchers. So the Gardner Awards celebrate the best in biomedical and health around the world. Dozens of their laureates in Canada have gone on to win the Nobel Prize around the globe. And over the last 2 years, we've had the real privilege of getting to showcase and celebrate some of their really, really spectacular researchers. Now, today is no exception. I'm really excited to be welcoming for the very first time today the Early Career Investigator Award winner, Anna Blakney. So she is at the School of Biomedical Engineering at the University of British Columbia, right down the road from a lot of our classes today. And she works with an amazing group of engineers, immunologists, and molecular biologists to learn how to prevent and treat disease. And more excitingly, from a science communicator's perspective, she really works to make sure that the public in general understands the scientific process, all sorts of things to do with vaccines in particular. And I'm really excited that she'll have the chance to feature both of those stories today with us. So without further ado, Dr. Anna, welcome to the broadcast, and thank you so much for joining us.
Thanks for having me. I'm so excited to be here.
Right. I know you've got a lot to share, so if you want to dive into your presentation, you are good to go. And welcome to our YouTube groups too. If anyone wants to identify themselves in the chat while Anna's pulling up her presentation, we'd love to hear from you there as well. Awesome. All right.
All right. Can you see my screen okay?
We can indeed. You're good to go.
Okay. Thank you. Awesome. Well, yeah, I'm so excited to be talking to everybody today. It's great to be here. So thanks so much for the invitation. So today I'll be talking about a little bit about our research about next generation RNA vaccines and therapies. Okay, so just to give you an outline of what I'm going to talk about, I think it's always fascinating to hear how scientists ended up in their roles. So I'll talk a little bit about my career pathway. I'll talk about our research in my lab here at University of British Columbia, and then a little bit of theoretical, just thinking about the future of RNA medicine and where we're going with this exciting technology. So just to give you an introduction, I did my undergrad degree actually in chemical engineering at the University of Colorado, which is where I'm originally from. I then went on to do my PhD in bioengineering at the University of Washington, which is in Seattle, so just right down the road from Vancouver, where I am now. I did my postdoctoral training, which is the training you do after you finish your PhD, and you kind of want to learn something more than what you did your PhD in. So I did this in an immunology lab, so very different than the engineering training that I had done. And I did that at Imperial College London in the UK. And then finally, I'm now at University of British Columbia. So my laboratory is in an institute called the Michael Smith Labs, and I teach in the School of Biomedical Engineering. So one thing I wanted to point out to everybody that people may not appreciate is that science is like a passport. So it's really amazing. Science unifies, everybody all over the world is participating in science. And although there's some differences in the way that people do things and the funding, it really is quite a unifying field. So I always thought that I would end up in Colorado just because that's where I'm from, but I've gotten to live in a lot of amazing places that, science has really opened up the door for me. So I've done internships in Boston and Pittsburgh. I did part of my PhD in Cape Town in South Africa, as well as Seattle, and then obviously got to live in London, and I'm now in Vancouver. So if you're interested in seeing other places, being a scientist is a great way to do this, either for where you work or even for conferences. So I think this is a really unappreciated aspect of science, but something that I really enjoy. So another thing, so we work in really what's called the immunoengineering field, and this really came to be from kind of my training in two very different fields. So I trained as a chemical engineer, and if you don't know much about that, chemical engineers really think about scaling up chemical processes, so how to make these bioreactors. Most of the time people go into like oil and gas fields, but my degree actually had more of a biological twist to it. So it's really quite a different combination to take chemical engineering and then do training in immunology. So immunology is obviously a field of biology that's all about the immune system. So it's very different from these very equation and math heavy chemical engineering field, but is also super important. So in a way, immunology to me is like learning a new language, but it's really been cool to be able to combine all of this training that I've had into this field of immunoengineering, which is really thinking about, okay, how do we make these new types of medicines that will hopefully improve the lives of people all over the world. So my point here is that I think it's really cool and especially important nowadays to get kind of interdisciplinary training and not to put yourself in one silo. So even though I'm like mainly an engineer, I also now work with a lot of immunologists, and a lot of our research is in the immunology field. So I think it's really inspiring to be able to interact with, and important to interact with, both these groups of people. So to give you a little insight into what my lab actually studies, it's obviously been a really exciting time in the field of RNA vaccines in the past few years. And I like to say that the plane is off the ground. So we now have 2 approved mRNA vaccines, which has been, I think, a very positive outcome of the pandemic. So where do we go from here? What do we do with this technology? So one of the things that my lab does is RNA molecular engineering. So this just means designing better RNA so that we get more protein expression from it, or it expresses only in certain cells, or it's more stable. We also work on biomaterials design. So the point of this is getting the RNA into the cells and tissues where we need it to be to have its function. And the last aspect of my lab is thinking about how do we make better vaccines and other applications of RNA technology, which I'll talk a little bit today as well. So I like to think about and really motivate our research by talking about, why do we even need new types of vaccines? We have lots of approved vaccines that we've been using for a number of years that are all really effective. So why do we even need to create new ones? So I just want to point out that there's still a lack of efficacious treatment for many devastating infections, such as HIV. In recent years, we've seen the emergence of multi-drug resistant bacteria. So vaccines could be a way of combating this. We're always looking to improve the safety profiles of already licensed vaccines. One that I think we can all relate to now is disease outbreaks. So how do we make a vaccine quickly in the event of a brand new pathogen? We also need to consider the changing age structure of the population. So as people are living longer, it's actually more difficult to immunize elderly patients. And so we need to come up with new vaccine strategies for this, as well as looking to reduce the cost and time required to produce vaccines. So the work that I do really is on for disease outbreaks, as well as reducing the cost and time required to produce vaccines. So for those of you who aren't familiar, RNA is a really fabulous kind of modular platform, we say, where as long as you know the protein that you need to encode, you can make a new vaccine for any disease really quickly, which is exactly what we saw happen in the COVID-19 pandemic. So the way that we make an RNA vaccine against a brand new pathogen is actually pretty straightforward. So this is an example for SARS-CoV-2. So there's a protein on the surface of this virus that's called the spike protein, which many of you may have heard of. We take the code for that protein and we encode it in our RNA. And it's a very fancy sounding form of it, which is the pre-fusion stabilized spike antigen, which is just a fancy way of saying the protein on the surface of this virus. We encode that in our RNA. We then have to put that into particles to get it into cells. So RNA is a large negatively charged molecule, and it won't get into your cells just by itself. So we have to put it in really what is a little fat bubble. So it's made of lipids that helps it get taken up into cells and also protect it on its journey there. So we then inject that intramuscularly into mice for our preclinical studies or humans for our clinical trials. So we've seen some really amazing data from all of these mRNA vaccines from the COVID-19 clinical trials. And this is can be a very intimidating graph, but I'll walk you through it. So this is some of the efficacy data from the Pfizer and BioNTech mRNA vaccine. And in this graph, they are comparing a placebo group, so a group that didn't receive the vaccine compared to the group that did receive the Pfizer and BioNTech vaccine. So this was from one of their original clinical trials looking at the efficacy. And their conclusion from this was that a 2-dose regimen of the Pfizer and BioNTech vaccine resulted in 95% protection against COVID-19. And this is the type of data that clinical trial people love to see, because there's a visible difference that you can very clearly see without any sort of statistical analysis between the placebo and the vaccinated group. And so they also concluded from this paper that the safety over a median of 2 months was similar to that of other viral vaccines. And this is the data that supported this being approved by health regulators around the world. So, these vaccines worked really well and made a big difference during the pandemic, but I think they can continue to be improved even further. And one of the reasons for this is because of the side effects of these vaccines. So I don't know if anybody's gotten an RNA vaccine. I know on the second and fourth one I had, I felt terrible afterwards. And so we're still really understanding why that is. But one of the things we've seen from these clinical trials is that it's directly correlated to the dose of RNA that you get. So this is also a lot of data. This is from one of Moderna's clinical trials where they were looking at the safety of their COVID vaccine. And in this, they used 3 different doses of RNA, so 25, 100, and 250 micrograms. And then they quantified the side effects that everybody had. So they grouped these into mild, which is gray, moderate, which is blue, and yellow, which is severe side effects. And again, this is really easy data to interpret because you can see as the dose increases, the number and frequency of the severe side effects also increases. So what we know from this is that we really need to prioritize minimizing the dose of RNA so that we can use these and not have as many side effects. This will be especially important for non-pandemic contexts, such as getting an influenza vaccine every year, as well as other applications of RNA. So the work that my lab does is using a special type of RNA, which is called self-amplifying RNA. So this is just a type of messenger RNA that's able to make copies of itself once it gets into a cell. So I have a very simplified schematic here below that I wanted to introduce you to. So for messenger RNA on the right, so this is what's in the COVID-19 vaccines. Once a copy of this gets into the cell, it engages with the ribosome and you get translation of your antigen. So this is the spike protein. This is what you make an immune response against. So for self-amplifying RNA, it starts out in a very similar manner. So a copy of the RNA gets into your cell, it engages with the ribosome, and again, you get some translation of your antigen. The saRNA has another special component though, which is called the replicase. So this is an enzyme that's able to just make copies of RNA. Think of it as like a Xerox for RNA specifically. So the replicase then engages with this original strand of RNA and starts making copies of it. So I've only put 3 here in the schematic, but it's actually thousands of copies. And thus you get a lot more protein expression from the same initial number of RNA molecules in the cell. So what this means is that we can actually use about 100 times lower dose of saRNA compared to mRNA, which should also hopefully then alleviate some of these side effects that we're seeing with these vaccines. So this is the type of RNA that I work on. And while I was actually still a postdoc at Imperial College, I had the privilege of working on a vaccine that was tested in a phase 1/2 clinical trial in humans. So I wanted to give you some insight into what that experience was like and just how amazing this platform is. So one of the most amazing things about RNA vaccines is the timeline of being able to make a new one and then get it into human clinical trials. So I wanted to walk you through anecdotally just the story of how our team was able to make our vaccine and how quickly that happened. So on the 10th of January 2020, the circulating strain of COVID was sequenced and made publicly available. So anybody could download the sequence and then start working on their vaccines. So our team at Imperial accessed the sequence on the 20th of January. Very coincidentally, the next day I flew to Vancouver to interview for my current position here at UBC. So as you can imagine, it was a very busy time. We ordered the DNA parts of our vaccine on the 22nd of January. We were able to construct our plasmid, which is the basis for making RNA, on the 28th of January. On the 12th of February, we confirmed that our RNA vaccine was expressing the protein that we expected. And so the next day, we started our immunogenicity studies in mice on the 13th of February. So this is when looking to confirm that we were getting the immune response that we wanted. We then started our rat toxicology study on the 30th of March to make sure that there were no safety concerns. We submitted our paper for the preclinical work on the 22nd of April, and then we started our human clinical trials on the 19th of June. So even as an academic group, we were able to go from designing a brand new RNA vaccine in January and starting our human clinical trials within 6 months. And to put this into perspective, people have done it even faster than this. So Moderna reportedly did it in just 66 days. So this technology really enables us to make new vaccines in a time frame that previously just wasn't possible. So everybody always asks where the Imperial vaccine is now. So when I moved to Vancouver in January of 2021, the government already had access to 2 different COVID-19 vaccines in the UK. And so they didn't want to fund the phase 3 trial for our vaccine, which makes sense. It would have cost another £130 million to do so. And so they thought those resources could be used better in other areas, which I think is very valid. So our team is now working on targeting COVID mutations and booster doses. I'm co-founder of a company that is developing this saRNA technology for vaccines called VaxEquity. And last year, we announced that we have a partnership with AstraZeneca to further develop this technology. So it's a really exciting time to see this technology coming online. I also wanted to point out that actually just a few months ago, the first saRNA vaccine against COVID-19 was approved. And this was made by a company called HDT Bio, which is based in Seattle, and this was approved in India. So just to put it into perspective, the dose that they're using for their vaccine is just 5 micrograms of RNA compared to the ones that are already approved. So in Moderna's, this is 100 micrograms of RNA, whereas for the Pfizer and BioNTech vaccine, this is 30 micrograms. So it's a much lower dose, which is really exciting. So now I want to talk about where do we go with this technology and why is it so advantageous for not just vaccines, but for many other types of medicines. So I don't know if you know a lot about how we actually make vaccines, but we use a lot of archaic technology for the current vaccines that we have. So here on the left, I have a picture of making the flu vaccine in 1957. So this was done in eggs. And this is actually a picture from 2021 of also making the flu vaccine, which is still done in eggs. So this is a highly manual and time-intensive process. Because of this, we have to decide what we're putting into the flu vaccine in February of each year for the Northern Hemisphere so that these vaccines are ready in October. And in that time, flu continues to mutate and change, so it's not always the most effective vaccine. Other vaccines we make in these huge bioreactors, and this is because we have to grow cells, which then either produce a virus or a protein that we purify out. And so again, it's really time and resource intensive. RNA, on the other hand, is made completely synthetically. So all we do is dump all the ingredients into a test tube or a very small bioreactor like this, and we're able to make 1 million doses in a volume of just 100 milliliters. So this is a really scalable technology, which is really why we've seen so many produced during the pandemic. So just to put this into perspective, I like to ask how many Olympic-sized swimming pools would it take to vaccinate the world with an RNA vaccine? So I did these calculations for the Pfizer and BioNTech vaccine. So assuming that there's 7.7 billion people in the world, which is what it was when I did these calculations, although I saw recently that we're up to 8 billion people now. So everybody initially needed just 2 doses. So this equals 15.4 billion doses total. Each dose is 0.3 milliliters. So this is a total required volume of 4.62 million liters. Each Olympic-sized swimming pool is 2.5 million liters. So this means we'd need just 2 Olympic-sized swimming pools to vaccinate the entire world with an RNA vaccine. And so this is a huge amount of liquid, but also doable, which is why we've seen now that billions of people have gotten RNA vaccines. So I wanted to talk about the manufacturing of vaccines, which may sound like a very uninteresting topic, but I think this is a real game changer for how we make medicines in the future. So I wanted to walk you through how we made our RNA vaccine for our clinical trial in the UK, and then how I think this is going to change in the future. So for our clinical trial, we got our RNA from California. We got our lipids actually from a company in Vancouver called Acuitas Therapeutics. Those 2 components were shipped to Austria and put into those nanoparticles that I mentioned. The formulation was then shipped to Italy to be put into the special vaccine vials and then shipped back to the UK to start our clinical trial. So even though it's a pretty straightforward synthesis process, none of it was done in the UK for our clinical trial, but I think this is going to change. So I can envision a world where instead of this, we don't need these centralized manufacturing centers anymore. So actually each country or region could have a manufacturing center where they make, not only, COVID vaccines or flu vaccines, but any of the other applications for RNA, such as personalized cancer vaccines or any sort of immunotherapy as well. So that brings us to uses of RNA beyond COVID-19 vaccines. And this was actually taken from the BioNTech website, which was who partnered with Pfizer to make the COVID vaccine. So they're one of the leaders in the field. And you can see their vision for RNA and all the things this is going to be used for as well. So today we have the Pfizer and BioNTech vaccine that's already available. Tomorrow they envision using mRNA for infectious disease vaccines, such as the flu, mRNA cancer vaccines, CAR T-cell therapies, as well as systemic immunotherapies. And in the future, this is really just starting in the field, but other applications such as autoimmune diseases, allergies, inflammation, and regenerative medicine. So there's really a whole range of applications that RNA can be used for. So another thing I'm really passionate about is science communication and really just talking about the research and helping to educate people about this technology. And so one thing I learned during the pandemic and kind of came to this realization is that if nobody wants to take the vaccine, then there's no point in making it. So as scientists, I think we have just like this elevated view that, we're going to make this vaccine and everybody's going to be excited about it. But that's not always the case, as we've seen during the pandemic. So this is a study that was done by a woman named Heidi Larson, who has studied this concept of vaccine hesitancy actually for decades now. And I think these maps are really insightful. So there's 3 different ones here. The text is small, but I'll walk you through it. So the first one says vaccines are safe. And there's a spectrum from people who strongly agree, which is darker blue, and then those who don't strongly agree, which is more the orange red. So the first one is vaccines are safe. The second is vaccines are important. And then the third is vaccines are effective. And you can see here that most people strongly agree that vaccines are important, but there's not as much agreement about whether vaccines are safe and whether vaccines are effective, although we have great data, especially for the COVID vaccines, to show that's true. So this really got me thinking, where does vaccine hesitancy stem from? And what is this missing link between believing that vaccines are important, but not safe or effective? So this is why I think public engagement is important. So talking to people, this can come in many different forms, talking to people on the media, using social media, even just talking to the people around you, your friends, family, and community. But why this is important is because the pace of science is just not matched by the pace of public communication. And so to put this as an example, as of January 2020, there were a lot of scientists that didn't know what an RNA vaccine was. And now billions of people have gotten them. So there's just this gap between what the technology that people know about and it actually being deployed. And this was a pretty extreme case. It did come online very quickly, and so just didn't give people a lot of time to really understand it. So because of this, I now run a TickTock channel that's dedicated to educating people about vaccines. And so I always say it's not just a platform for dancing teenagers, although there are a lot of dancing teenagers on there. So I started this in October 2020 as part of an organization called Team Halo. And this was started as a collaboration actually by the United Nations and the Vaccine Confidence Project. And the whole point was to get scientists and clinicians who are working on COVID-19 on TickTock specifically to be able to answer people's questions. And I think it's actually a great platform to show people how we make and test vaccines and also directly confront misinformation. I think it works because the videos are really short and engaging, and it offers a palatable amount of information. So you really have to boil your message down to 15 or 60 seconds, as opposed to, a lecture like this, like scientists are used to giving, where you either get, 30 or 60 minutes to really delve into the topic. So it really forces you to have your message that be easily understandable by people. So one of the things I love to do on my TickTock channel is also recognize the scientists who are behind the COVID-19 vaccines. So here I have two different people who have done years and years of research on this technology. So the first is Bob Langer, who's a professor at MIT and who's a co-founder of Moderna. And then the other is Peter Cullis, who's actually a professor right here at UBC, who really developed the lipid nanotechnology that made the vaccines work. So he started Acuitas Therapeutics, which is the company that formulated the Pfizer and BioNTech vaccine. So that came from right here in BC, which is really exciting. And although there's definitely a lot of trolls and negativity on TickTock, I think what's really encouraging and exciting is you can see in this comment section here for Peter's video just how appreciative people are. You can see people saying, thanks for saving the world, real life superhero. Thanks for saving lives. Thanks for all your hard work. And this made me realize that people really appreciate scientists, but they also have no idea who they are. So part of my goal is to make more scientists household names. So in conclusion, with the state of RNA vaccines right now, we've seen that these mRNA vaccines have enabled a rapid response to the COVID pandemic. We've seen that RNA vaccines are made synthetically and thus require a smaller footprint and timeframe to make than traditional vaccines. We've seen that the side effects have been shown to be proportional to the dose of RNA, and thus minimizing the dose of RNA is a real priority. But also that communicating to the public about science is really imperative so that everybody understands this technology and is able to ask questions and get them answered and make an educated decision for themselves about whether they want to get a vaccine. So with that, I'd like to acknowledge all my team, so my current lab members as well as alumni and our collaborators at Imperial College and Precision Nanosystems, as well as Acuitas, our funding sources. And with that, I'll take any questions. So on this slide, I just wanted to showcase also how fun it is to be a scientist. So this is a picture of all the people in my group. This was on our lab retreat recently. So we went to a place called Loon Lake here in British Columbia and went canoeing and did lots of hiking around the lake there. We're also big hikers. So this was up to the Lions just outside of Vancouver. And then our holiday party just the other day, we went go-karting, which is very Formula One inspired, but it's really fun to be a part of a lab, and scientists are generally a very fun group of people, which I think is also a well-kept secret. So thanks so much, and I'm happy to answer any questions.
Fantastic. Well, Anna, that was a spectacular presentation. If you want to come out of screen share, see us again, we can have a bit of a conversation. I want to ask first, did you win go-karting? And no one's
I did not.
No?
No, oh. my students are ruthless, so they didn't let me win.
That's a bummer. I mean, you can kick the really bad ones out of the lab, I guess. You have a little power over them. We're going to give our students just a second to let those questions percolate. So, Mr. Lee, I will come to you guys in a minute. YouTube groups, feel free to share in the chat, but I'll share some of our written in ones from classes that couldn't make it live today, but are going to tune in later. So the first is a question we always get, every program universally. Do you have a favorite part of your job? Now, you spend a lot of time talking about the collaborative nature of science, that you get to do amazing things outside of just doing scientific work. Is there anything that really stands out on a day-by-day basis of your work?
Yeah, I guess my favorite part of my job is really just, I feel extremely privileged and grateful to be a scientist, because what I love about science is being able to think of an idea and then actually go in the lab and do it. So that's what I think is so inspiring, and just, it's really, it's satisfies my curiosity, I guess. But I love just thinking about oh, maybe we could do that, or oh, maybe that would work. And then actually getting to do the experiment and see how it goes.
At the beginning, you shared this great career trajectory and you went to all these different places, all these different sort of fields of inquiry. Now, did you have like a plan in mind? You're like, okay, I could get those things together and it would lead to something great? Or were you doing a program and you're like, oh, this, part of it really speaks to me and I'm really excited about it. And then you end up going into the new field. Like, how did that progress for you?
Yeah, no, I did not have a plan for all of this. It wasn't like I had the foresight to know that it would go exactly on this trajectory, but I would say it was more just opportunistic. So whenever you're at a university or in a program or just wherever you're working, oftentimes there will just be opportunities that come up. And that's really what I did, was just take opportunities that were given to me. And some of them were scary. It was scary to like move to South Africa by myself, but it was such a cool experience. So I think having those challenges and opportunities is a really cool aspect of this career.
One of the things that I always like to highlight with scientists is that, the idea, if you're a grade 8 student, like our class today, for instance, live with us, the idea of 4 years of school and then 4 more years of school and then a few more years of school and then a postdoc is really daunting and scary. But as you do that process, you get the chance to have experiences like you were talking about, and you end up with a career where you get to play for work and you get to have a lot of fun and work with amazing people. And I really think that's an important thing for students to think about.
Yeah, I agree. Sorry to interrupt you, but just to add on to that too, I think if you like frame it like that, like, oh, you're going to spend so much time in school, it does sound really intimidating, but for sure, grad school and postdoc is very different. It's more like a job. So you're not like in classes all the time and taking exams. It's more like you're working as a researcher in a lab. So I think that's really important to keep in mind, is that it's just a different type of school. It's really like you're trying to learn how to become an independent researcher as opposed to majoring in a certain field and getting a degree in that.
Fantastic. Anna, we're going to have Mr. Lee's class come in a second. So Mr. Lee's group, if you guys want to unmute your mic, I'll come to you in 2 quick seconds. While you're unmuting, I'll just note, it's so funny to me that you came from Colorado. I don't know why this is the case, but having done over 1,200 broadcasts, American scientists that are super cool come from one of 3 states. It's Colorado, it's North Carolina, and it's Arizona. I don't know why that is, but it's like a universal. It's amazing.
That's a really interesting observation. Someone should look into that.
Yeah, there's something about the culture. I don't know. Mr. Lee's group, coming to you guys for a question. Come on up. Hey, guys. Hi. Does anyone have a question for Anna? I know we're in grade 8. And it's very challenging to ask questions sometimes.
I see some hands.
Are there hands?
Yeah, we just had a question for Anna here. Wondering if at what age, when did you decide you wanted to become a scientist?
Cool.
Yeah, that's a good question. I don't know that I ever made like a conscious decision that I was going to be a scientist. So what really got me interested in it was actually I visited a university, actually University of Colorado, where I ended up going. While I was in high school, I went to like an engineering kind of like day camp thing there. And that was the first time, like, I just thought engineering was like, actually my dad was an engineer, but I thought engineering was like super boring. Like I thought it was just people that did like math and like made drawings of things. And I don't know, just had like super boring jobs. And I went to this camp and it made me realize that engineering is really cool. Like all the things around us are like designed and made and tested by engineers. And so that was really empowering. I remember one of the professors talking about actually diapers, which is something that just seems like so, I don't know, uncomplicated and straightforward. But she was talking about how actually like the polymers used in diapers were made by biomedical engineers to absorb more liquid. And I was like, wow, it hadn't occurred to me that like everything around us is made for and optimized by engineers. And so that's what I found so inspiring about it, which is what prompted me to major in engineering. And then I worked in a research lab while I was in college. And that was just, I thought, yeah, I just found kind of what really worked for me. Like I loved being in the lab and doing experiments and really seeing how you apply your learning to actually doing science and experiments. So I kind of just became a scientist. It was never like my ultimate goal.
But I love that you had this amazing opportunity. And this is something that we like to highlight with students a lot, is that if you ask scientists for advice and feedback, I mean, 9 times out of 10, scientists don't bite. Like they're really keen to share their passion and enthusiasm for topics and offer those sorts of opportunities. I remember seeing Saturn's rings through a telescope when I was like 14 years old, and you can see it 1,000 times in a book and you see it through a telescope and it blows your mind. So I'm really glad you had that experience. By the way, on the engineering note and material stuff, shameless plug, The Martian is the greatest engineering movie of all time. time. Like it's like what Jurassic Park did for dinosaur people. The Martian, just you do everything with duct tape and it's amazing. And stuff matters. This is a really fantastic book. Might be at the level of grade 8 and above, but it's like the best book on things like cool diaper technology, which is great. And yet I never thought I'd say on one of these broadcasts. So thank you for that segue. Anna, you shared this great image of the world map of people being vaccine hesitant, feeling that they're safe and effective. And it's so interesting seeing the very regional differences there and sort of outlier countries. For people that are looking to find reliable information about vaccines, of course, they can come to your TickTock channel, they can follow you. Is there a general advice you'd give for resources or websites or what have you that are accurate and up to date for vaccine and general medical information?
Yeah, so I would say, I guess there's like kind of two aspects to this. So, the government of Canada does a really good job of keeping all of the information about like vaccines or different medicines up to date. And with what's really important is having links to where that data comes from. So I know not everybody is like me, where I've received so much training to read these journal articles and be really critical about them. But one of the other things that I've really come to realize during the pandemic is we get information without knowing it from sources all the time, right? So you see it in the media, you see it on social media, and one of the things that I try to do on my TikTok, and I think this is something really important to do, whether it's the media or social media, like anywhere you're getting information about vaccines or different medicines, is know where the data comes from. So I always try to put a link to the data and make sure that you can actually trace it so you know where it's coming from. So is this a journal article or is this just some press release from a company? That the quality of data does vary. So wherever you're getting information from, I would just say, make sure you know where that data originally came from. And that's how you can gauge if you can trust it or not.
Yeah. My wife's a librarian and has recently been doing sort of disinformation and misinformation programs for students at universities. And one of the big things to note too is if an article has a sort of inherent bias, or if they're trying to get you angry and riled up, people know that that's a way to engage people. And so you see that on a lot of social media platforms. You see that from a lot of sort of ideologically based people trying to convey information to you. So look for the source, how they're conveying that information. And I love the idea of tracking down the data whatever you might be looking at to get that original stuff. Mr. Lee's class, we're coming back to you. We're going to take a couple more questions, then we're going to wrap up because time flies when you're having fun in these programs. Back to West Kelowna. Hi, guys.
It's good.
What's your question? Nice and loud.
What's your favorite book, Anna?
Yes, so there's a book that I love and absolutely recommend to everybody. So it's called The Emperor of All Maladies, and it's written by Siddhartha Mukherjee. He's an amazing author for anything science related, but I would say he's a clinician. He's amazing clinician, so he treats people, knows everything about these diseases, but he's also kind of a historian. So he walks you through, cancer, how it used to be viewed, how we've treated it, how we treated it over time, and kind of where we're going. But it gave me just a more comprehensive and understanding, even from all the training that I've gotten as a biomedical engineer. So he has a couple other books out, The Gene, and a brand new one as well. So I cannot recommend him enough. Nice. It's like this was planted.
I know. Well, I looked for Emperor of All Maladies, and it's not on this bookshelf. It's on my other one, but spectacular. Have you read Song of the Cell?
I haven't read it yet. This is my holiday plan.
Okay, it's coming to me at the library soon. But yes, it's like the golden age of science communication. You're a perfect example of this. Social media platforms that didn't even exist a few years ago are now sort of the main means in which a lot of people get this information. But whether it's books, podcasts, online art, science art has become a huge deal. It's really exciting if you want to be entranced by all manner of scientific topics. So I'm so glad you mentioned him specifically. He's a great author. Anna, okay. We're going to wrap up with, two quick questions. CRISPR and artificial intelligence are two things that are sort of revolutionizing a lot of medical fields. They're getting involved in a lot of, they're changing everything in a really rapid way. How do they relate? This is a broad question to your field. Are they speeding things up? Are there risks associated with them? Anything you want to share with us? Because I know they're being talked about in some of our other Gardner programs.
Yeah, absolutely. So there's definitely a lot of overlap between kind of what I do with RNA vaccines and therapies and CRISPR. So CRISPR is a way to edit genes, right? And so you can actually do this with RNA. So we actually have a brand new project that we're working on now to encode CRISPR in our RNA and be able to edit certain tissues that way. So similar similar, I think, CRISPR and RNA have had similar moments in the past few years or so. So they're both really exciting technologies. But one thing I think it's good to just be realistic about these technologies. Like, I don't think CRISPR is going to fix every genetic disease and RNA is going to become every vaccine overnight. They're really exciting, but they have very certain applications where I think they'll be effective. So I think that's just the important thing to consider. And then AI is also super important. So now that everybody uses like sequencing to see, okay, what's happening inside a cell when we introduce RNA or we introduce CRISPR, those data sets are enormous. So you just get these all these like millions of letters of sequences, and you have to decode that and know like what's happening in a cell and then what's happening in all the other cells around it. So AI is a great way to handle these big data sets and kind of get to a level of understanding that you couldn't do just looking at it as a person scrolling through all of these sequences. So this is a really important tool for us to handle these big data sets and get insight so that we can design even better vaccines and therapies.
You are really good at taking a question and putting it into concise and very fantastic. Very good. Cool, Anna. We're at the 40-minute mark, so I want to note again, this is part of our Gardner Award series. You can check out Anna's recognition there and some of the other scientists that we're featuring all week long and into early next week. If you want to see Anna on TickTock, you can check her out at the link below. And if you want to dive in with the science, we've got a UBC link here, which isn't actually, this is like the least unwieldy URL of all the links of scientists we've had in a long time. So way to go for making even that concise and nice. Anna, before we wrap up, one last question. Is there any final message you'd like to share with our students today, joining in live after the fact? How can they learn more, be excited? What would you like them to leave with about you and your work?
Yeah, so if you have any questions, like feel free to reach out to me. Like I love talking to people about science. One thing I always mention, and this is really like what motivates me to be on TickTock, is that I think we have this idea of vaccine hesitancy, and we kind of class people into like pro-vaccine or anti-vaccine. But I actually think the majority of people fall in between there, and vaccine hesitancy is a good thing. We actually want people to be questioning what they're putting into their bodies. And so I think the challenge there is really just being able to ask questions and understand more about these technologies, which is why I'm on TickTock, being able to do that. But I think we need more scientists on TickTock. So that's really always something that I'm encouraging people to do. So, yeah, if you have any interest, feel free to reach out for me. I love hearing from people.
You actually have a very easy, publicly accessible email, so people can reach out to you quite readily if they're so inclined. Anna, this has been so much fun. Thank you so much for joining us today. It's been a really special part of our Gardner series. As we do to end every broadcast, I'm going to bring in our class to say thank you and farewell with me. But Mr. Lee's class, if you guys want to unmute your mic, we are done with the broadcast. Thank you so much, everyone, for the great questions.